see my screen.
Perfect. Now go ahead.
So you've got the screen so hello everyone. So it's a great pleasure to be invited to give this talk at the EPNOE webinar series. I think it's really a great opportunity to connect with everyone across the people who are very enthusiastic about polysaccharides. So how to put it together in the best possible way. This talk will be slightly different probably from what normally people are, how the people present this their work. So I will try to go into really fundamental details after I've introduced some of the opportunities that we've been exploring with a polysaccharide from Plantago ovata. So, but without going ahead too much, I will start my presentation just by introducing myself. So I'm leading a team in the University of Nottingham in the School of Biosciences. They call this Automated Biomaterials and Biointerfaces. And we are the team of enthusiastic and like-minded people who are interested in different aspects of the carbohydrate physical chemistry. And we're also developing a number of the capabilities to characterize nanoscale and macroscale flow and gel properties. So we are working also with a number of industry partners because as Tina already and Marie already mentioned, that I have these both backgrounds in industry and academia. And in essence, in a very fundamental aspects, we're interested in the compolymers and all the polymer brushes. And a lot of polysaccharides are actually exist in the form of a polymer brushes. And this is our sort of particular area of interest. So in my group, we have several research areas associated with the dietary fiber functionality, and especially those dietary fibers that come from complex plant cell wall assemblies, as well as we kind of branching, we're learning how plants, I will point the option laser pointer. Can you see my laser pointer so that it will help navigating through the slides if they're busy?
Yes, I can see.
Fantastic, Tina. This is brilliant. So complex polysaccharides is the one area which I think targets a very clear impact improving the diet in this changing world. The branching from this one, we have a theme on the polysaccharide gels and composites where we try to develop the new materials either for biomaterial applications or for the packaging applications. Recently, we've started this type of work. We also work actively on the mucin glycoproteins, which are carbohydrate heavy proteins, and we're interested in their structure on the mucosal membranes, as well as the role of mucin in the oral processing and the saliva product interaction, which is very important for the food industry. So in addition to our sort of research interest in scientific area, we are also developing a number of capability, especially in the area of the nanoscale characterization and atomic force microscopy. So we've developed techniques to analyze mechanical properties of complex structures, as well as analyzing adhesive interaction in the nanoscale fibrillar networks, where we can measure the adhesion between the nanoscale fibers, which is probably a topic for another talk, how you can measure nanoscale adhesion. And we've applied these for the cellulose networks, which would be very instrumental for a lot of you probably on the other side of the screen. So this is just a big advertisement a little bit. Taking the opportunity that we've got funding for this biofluidic microscope that will be commissioned early next year. So it's all been ordered and procured. We're just commissioning this instrument, and it combines essentially a high resolution confocal microscope with the Fluid-FM atomic force microscopy system that enables you really to perform things not only related to use of the atomic force microscopy under the optical guidance with the high resolution and high speed acquisition, but also do the microfluidic experiments where you can inject something in the cell, or you can sample something, or you can manipulate the objects, including like bacterial cells or mammalian cells, as well as do the engineering applications like nanoscale 3D printing. So this instrument is coming into our labs, and if you have interest in this type of opportunity, please give me a shout. Please do email me and get in touch, and we will definitely discuss what are the opportunities for collaboration there. And it also designed for a lot of awkward systems, because we have a huge positioning range of the more than 100 micrometers in all directions, which enables us to put quite challenging sample, not just the typical AFM mica and everything is flat. So this is all designed with the biological and systems that try to be 3D as much as they can. So I will try to start today's lecture with introducing plant hemicelluloses and xylane biosynthesis. So when I was in University of Queensland, I was the part of the Australian Research Council Centre of Excellence in Plant Cell Walls, and we were interested in xylane biosynthesis. And because xylane biosynthesis is responsible for production of those plant hemicelluloses, have a really a key role in the structure of the plant cell walls, which are part of the also food chain, because it is a dietary fiber, is a fodder for animal, commercial animals. So these cell wall structures, they're made of the cellulose, but also the hemicelluloses there are to link and to mediate this interaction between the cellulose fibers. And so we were interested in how the cellulose biosynthesis is performed in plants. However, and of course, we're interested in those plants that have a commercial interest, for example, wheat or corn. Unfortunately, in both wheat and corn, the biosynthesis is quite complicated, and doing the genetic analysis is quite tricky. So we thought we need a model plant, and it's very typical thing in the plant sciences when you need to find a model plant. If you can't, you like Arabidopsis. So first you do the work in Arabidopsis, and then you try to do work on a different plant, like barley, for example. And we've chosen a whole sort of genus of plants called the Plantago genus, and different species within this genus. One of these is a Plantago ovata, which is very well known xylem, and you can also probably recognize different species like the Plantago major. All people in, for example, if you're somewhere in Scandinavia, you can definitely recognize those. And if you have, for example, Plantago lanceolata or Plantago ovata, if you live in the tropical areas, you can recognize those plants. So let me introduce you, like Alice in the Wonderland, the queen, EPNOE. This is the genus Plantago. Genus Plantago, these are your EPNOE members. So we were trying to use these Plantagos as a model plants for the xylane biosynthesis. And the reason why we were chosen them, because each of these Plantago species produces seeds that extrude these xylane-rich mucilage. So you can get a lot of material in a very short period of time. So in essence, that was one of the motivation why we went for the exploration of this as a model plant. And so it was absolutely fantastic. And we started to dig deeper into the genetics, and we realized there is a huge variety of xylanes that have been produced by different species. And essentially it was very stimulating, because essentially each species of Plantago, in terms of their production of the xylane, was akin to these Darwin finches, where each adaptation to each to every environment they're living in and they're growing is exemplified, and it's translated into the structure of this xylane. So if you have a lot of rain, you have one structure of this xylane. If you need to conserve water because you're in the arid climate, then you have a different structure of this xylane. You have the same blueprint, the same template, that then the variation in the composition and variation in the structure and the motif will result in a different properties and different interaction with water. So it was all very exciting, and we started digging deeper into the xylane biosynthesis of Plantago. And we thought that it's a fantastic model, because it is really nice. You can get easily sample. That's what we thought. But the reality was very kind of very odd, because Plantago actually does the xylan biosynthesis very differently to what wheat or corn would do. And for that reason, as a model plant, is not very good. And so at some point, we were kind of really disappointed because, right, it's a model plant and it's not a model of what we should be doing. But then we realized that actually Plantago mucilage has a lot of opportunities. So actually it's a super cool material and a super fascinating material that deserves its own place in the among the commercial crops. So one of the missions that we have is just commercializing this as a crop, and it's already used because it is a xylem fiber is known for a long time. It's been used and it's been underutilized and can be used more. So in terms of the xylem complex xylans, significance and opportunity, we can go from foods to packaging to biomaterials and fiber or drug delivery applications. So as I said, this talk will be slightly different to most of the talks where people will start talking about the fundamental insights and then go into the applications. I will just highlight a few applications of these xylem polysaccharides, and then we're going to dive deep into the physicochemical properties of these xylans, and then we'll try to find what is the unique underpinning mechanisms that are driving their particular functionality in different applications. So we have the application in foods, and here we can either look at the interaction with the starch networks or the protein networks. And obviously the starch networks is very useful for all type of baked products where we combine the xylem fiber with starch granule. Also the protein networks where we, for example, can try to utilize xylem fiber as a replacer for the thickeners for the meat analog application when you have the, for example, plant protein matrix, and you can utilize xylem to create plant analog products. So, for example, just to give you a little bit of flavor, when you look at the interaction of the fiber with starch, and in particular, for example, rice starch, we looked at the different starches, and the rice starch is just one that is very easy to show and highlight the effect of the fiber in this case. We were looking at the syneresis, so the separation of the fluid from the gel. And what we found is that if you use the xylem fraction, you can actually reduce freeze-thaw syneresis by a huge margin. So if you essentially put the rice starch into the freeze and you take it out several times, you will see a huge syneresis, condensation, retrogradation of rice starch, and that releases huge amount of water. And if you essentially take an opportunity to take a composite with the xylem fiber, you can reduce by an order of magnitude the freeze-thaw syneresis, as well as improve the overall syneresis properties of the composites. And also, if you see from the mechanical perspective, measuring the rheological properties. So now I'm just showing you these oscillatory spectra. So this is the large amplitude oscillatory shear. So when you go from the small shear, small oscillation to very large oscillation, so essentially breaking the gel. If we look at the rice starch, if you get the rice starch and then after the freeze-thaw cycle, you will get a huge consolidation of the material that is resulted from the expulsion of this water. However, in the presence of the fiber composite, even after 5 days of the freeze-thaw cycling, you can see that you have a conservation on the mechanical properties. I always joke about the sort of frozen lasagna that tastes as a fresh one. So in essence, it gives you an opportunity to improve the stability of the product, improve their freeze-thaw stability, which is of huge importance for the cold chain and sustainability, because obviously there is a huge energy spent on the storing thing cold. And also these fiber have an impact on the digestion, and we always were discussing what is the mechanism. Is it like a decoy substrate, or it kind of coats the starch granule? And we found that the primarily is due to the mechanism associated with the formation of this occlusion film, that the xylem can actually, the Plantago ovata fiber xylan can protect the starch granule from the attack of alpha amylase in saliva and in intestine, and thus reduce the glycemic response. You can also look at the use of the packaging of these materials in the packaging. We've been working with our collaborators in KTH, in Francisco Villaplana and Jenny Berglund. So we were looking at the interaction of the celluloses with hemicelluloses, and obviously in this case was mainly the spruce and birch. And we found quite a unique set of micromechanical properties that lead to a plastic-like response of the cellulose film. So you all know that, for example, if you put paper in water, it disintegrates. But if we can create our composites, they are not disintegrating in water. They can withstand water, and they can be used for almost a wide range of packaging application when you need the stretchability or you need a water resistance. So this is really an excellent opportunity to move us away from the use of plastics. And we started using our composites of xylans with the complex xylans with cellulose using the ionic liquid regeneration method, where we've shown the opportunity for these polysaccharides from the composite and from the hydrogels and films with the unique micromechanical and the hydration properties. So this is again just an illustration of why we should care about these materials. And finally, if you go to the fiber and drug delivery application, we're obviously very interested in fiber. I mean, Xylem and Plantago, which is the synonyms, are important dietary fiber, and we've proven that because it has a potential to deliver a lot of benefits in terms of the gut microbiota. So what we know is that the slow non-digestible fiber, they go down through the digestive system, end up in the colon, and that is where the microbes, they started fermenting this complex dietary fiber and essentially produce short-chain fatty acid, but also the gas, which is a byproduct of the fermentation reaction. And this gas that causes uncomfort, bloating, and that precludes people from using the dietary fiber as much as they should because of the negative sensation. And so we've collaborated with our friends at Quadrum Institute, with Fred and Hannah, and we've been looking at the fermentation of these complex fibers from the xylan. So we fractionated it and we looked at the behavior of different fractions. And what we could show is that we can increase the production of the short chain fatty acids per milliliter of gas produced. So this really gives us an opportunity to reduce this gas volume without compromising the production of the beneficial short chain fatty acids that help us to maintain healthy diet and healthy metabolism, because they go into the brush wall of the colon, and then they go into the bloodstream, and then they end up in liver, and they start contributing to the systemic metabolism, and that's a part of the very important part of dietary fiber functionality. Right, so I hope that I've convinced you that xylem can be effectively used in foods. It can be used as an additional material to cellulose in packaging applications. It can be used as a fiber for the microbiological or microbiota functionality, and also can be used as a drug delivery agent, but also can be used in biomaterials application and in tissue engineering application as well. So it's really a broad spectrum. So let's go back to our xylem. So you have this mucilage that is extruded from the seed, and I'm going to show you the video. I don't know how it's going to play because I don't know what will be the delay, but I'll try my best. So this is now the microscopy images of how the xylem gel is extruded from the seed. So you can see the seed, and then you can see that there is a stained mucilage. So if I press point option pen, no, point option. Oh yeah, so for some reason it doesn't do. So I press the play video, and you can see the expansion of the mucilage due to hydration. So obviously this is the speed up video. And you can see that when there is this expansion happening, is not a homogeneous material. And different colors are corresponding to different stains. So in this case, it is the chalcofluor white and direct red 23. And so you can see that there is a formation of the three layers. I don't know if you can see it quite well, but there is this diffuse layer on the outside that is primarily green, which is chalcofluor white. Then you have a layer that is kind of has a lot of red, which is direct red 23. And the third layer is primarily stained with the chalcofluor white. So it's primarily green. So you have this layered formation. So the mucilage is not a homogeneous material. And so with my PhD student, Dr. Long Yu now, we've investigated this multi-layer structure of the mucilage. So we've discovered this to be honest, by accident, that the different stains would have a differential staining of the mucilage. So we can see that there is this diffuse layer. I'll switch back to the laser pointer so you can follow where I'm pointing. So there is this diffuse layer that disintegrates, it's fully soluble in water. And then we have this gel layer, which is stained with it primarily by direct red 23. And then we have another gel layer that is stained by the chalcofluor white. So we've developed the protocol for isolating fractions from different layers. So essentially the outside layer was very easy. So you can just use the cold water to extract these polysaccharides. And when we've analyzed their structure, it is turned to be a conjugate of RG1, rhamnogalactorial pectin, and complex heteroxyl or arabinoxylan, because it's made of arabinose and xyloses. So it forms a viscoelastic fluid, or a low concentration, just a viscous fluid. At the high, it becomes the viscoelastic fluid. But then what was interesting, there were two gel fractions. The one gel fraction was melting at about 40-41 degrees. So you can extract it using the hot water extraction. And you use, for example, 50 or 55 degrees water to extract this fraction. And then what is left is another gel fraction that doesn't melt at even 100 degrees, and it can only be solubilized in alkaline solution. And we call these fraction the water-soluble gel fraction and the alkali-soluble gel fraction. So we've isolated these two gel fraction. One is melting at about 40 degrees, and another one is the thermostable. So we were really fascinated by these fractions. So we've did all the linkage analysis to look at the constituent polysaccharide, and we also did macromolecular characterization and the hydrodynamic characterization using sec moles and DLS as well, and static light scattering, to be honest. And we were really surprised. The first thing, there was a very high level of the backbone decoration. So the decorations is almost higher than 90%. It's actually more close to 95%. So only every 20th unit of the backbone is unsubstituted. The rest of them are decorated with the side chain. The second, as you can see, the huge is 1,3 xyloses. So the biggest decoration that we have is this 1,3 xyloses. And this is very unusual linkage in the plant kingdom. You can find them in the algae, but you can't find them in plant kingdom. And then what you can see that the molecular composition of this water-soluble fraction and alkaline-soluble fraction is almost identical. So exactly, you kind of say, okay, composition, monosaccharide composition is the same, but linkages are different. That would explain the differences in the gel structure. And here, the linkage composition is essentially identical. That you can't really, if you do the, we did a few replicates, we did a few batches, and the result was consistently the same. These fractions are, from the linkage perspective, linkage composition perspective, are identical. So this is just a representation of what would you get in terms of the structure based on the budget of the linkages that you uncovered. So it's highly substituted. And obviously the side chains is primarily anywhere from 1 to 3 monosaccharide units comprising of different combination of arabinose and xylose residues. So essentially two monosaccharide, arabinose, xylose, very unusual 1,3 linkages that link the xylose to the backbone. So these xylose side chains 1,3 linked. And you have this very unusual, very high level of substitution of almost 95%. So the question that we had, 2 polysaccharide fraction, what is the mechanism of the gelation of these fractions, why they're different, and how can we engineer new materials? So this is the question that we wanted to ask. So first we look at what is the mechanism of the gelation. So if you measure the rheological properties, you instantly start to see that both of these gels are not real gels. They are physical gels because you can see the dependency of the G' and G" on angular frequency, suggesting that these are not, in a full sense, true cross-linked gels. So there is some relaxation of the structure and the rearrangement of the structure going on. So this is already suggested that these gels are different from most of the gels that we know formed by a very strong junction formation mechanisms. There are obviously, because we are in the polysaccharide network, there are a lot of other things that you can link the structure to. Is the molecular weight. We already checked they're the same molecular weight. We also checked the ferulic acid content because people were saying, oh, it could be deferrylate crosslinks, and we've proven that it is not the case. People were looking at the methylation and acetylation. We've checked that, we've characterized that. That's also the same for both fractions. People were suggesting it could be some cellulose colloids that's been taken care of and been sort of proven that this is not a factor, and basically we don't have much of the cellulose colloidal or cellulose nanofibrils retaining in any of these gels. So obviously, to take now from the information that we have from the rheological characterization that these are physical gel, we said, okay, if they're physical gel, then if you put the hydrogen bond inhibitor, the physical gels should dissolve. And here's exactly what happens. You put these gels in guanidinium hydrochloride, and instead of a gel, you form a fluid. So that is a clear indication that the hydrogen bonding is a key mechanism for maintaining the gel structure. And similar effects can be induced by higher concentration of potassium hydroxide, or you can use urea or any other hydrogen inhibitor. So essentially, there is a hierarchy of the hydrogen bond. So you can actually form a different level of the hydrogen bond depending on the solvent qualities. So when you go to the non-gelling condition is where you inhibit formation of the hydrogen bond, and the gelling condition associated with the formation of these hydrogen bonds. So can we probe this hydrogen bonding? And the answer is, of course, yes. So in this case, we were investigating the rheological behavior very carefully as a function of the concentration and as a function of temperature. And here we came up with the system where we are looking at the changes in the G' that is linked in the number of the cross-link that you form within the gel network. And you look at the sort of essentially the percolation threshold for this network to form as a function of the concentration, obviously. And from that, you can extract the thermodynamic parameters associated with the formation of this junction. So this is also been supported by the more conventional time temperature superposition measurements. But the problem with the time temperature superposition measurements is that you can do it very nicely on this melting fraction wedge transition from the liquid to solid, but you can't do the same trick with the solid fraction because there is no obvious transition on the temperature. So the time temperature superposition gives you a very nice confirmation of this result, but it doesn't give you the full answer. So we came up with this way how we can essentially utilize this rheological approach as a function of temperature and use the Arrhenius plot and the Arrhenius model to calculate the thermodynamic parameters of these junction zones. And we have also kind of characterized, got the additional information from the large amplitude oscillatory shear measurements. So you can plot the Lissajous plots and look at the evolution of these links under the condition of the stress or rate. And you realize that these are temporary physical crosslinks that have a specific characteristic breakage and reformation time. And so essentially what was interesting is that for the different fractions, this behavior is absolutely totally different, because for the water-soluble fraction, you have a huge thinning effect. So as soon as you apply shear, there is a thinning effect. But for the alkaline soluble fraction, by contrast, you observe a huge thickening effect. So you start shearing it and it resists the shear. So essentially, the more you shear, the more the gel consolidates. So this is the totally different response to the shear and to the deformation. So essentially, the weak gel that is formed, that is your water-soluble fraction, and alkaline is a fraction that has these consolidating properties. And we could sort of proven it in the relaxation or step relaxation experiment, strain relaxation experiment, that enables us to determine not only the energies, which we determined from the Arrhenius plot, but the time stamp for the dissociation of these junction zones. And we realized that essentially for the water-soluble fraction, there is a single mode recovery with the characteristic time of about 20 seconds. But if you look at the, for example, alkaline fraction, it has a two-step relaxation. One is very similar to the water-soluble fraction of about 20 seconds, and another one at the order of magnitude of 10 minutes. So essentially, the junctions in that alkaline fraction relax in two ways. One is a fast relaxation, but then these consolidating properties are responsible for this longer relaxing fraction that are relaxing on the scale of about 10 minutes. And that gives an opportunity to engineer materials with the self-healing properties. So it means that we can bring these gels together and they will fuse, because the molecules still have sufficient mobility so that we can really observe this fusion between the gels, because these gels are not fixed in stones. There's still residual mobility. These could be very slow, but these gels still sort of have this opportunity to integrate with each other. And so you can use, for example, these materials for the 3D printing in a very advantageous, because you don't need to glue them. You just print one layer, then you print one layer, then you wait, and then essentially they will fuse into a single material. So the question next that we need to answer is, do different fractions form different gel structures? Because obviously we know that they're rheologically very different. And we actually look at the gel structure from different perspectives. So the one is, of course, the microscopy, but microscopy is always deceiving. So this is actually the cryo-SEM data made using the high pressure freezing to avoid the ice crystal formation. And they show that the differences are very minor. And of course, I mean, we can't trust microscopy, so we need to go to scattering. So here we used an opportunity of doing the ultra small angle neutron scattering and small angle neutron scattering, and that we could see a kind of essentially that the difference between the two are very small, and they're all at the very low Q. That's a higher level of the network arrangement. So at the nanometer and 100 nanometer scale, these gels are totally identical. It's just the higher level structure is slightly different between the two. And the same, what you can see, for example, does the changes in the structure are associated with the changes in the rheological properties? And we couldn't find any differences in the major difference in the structural changes associated with the changes in the temperature. So it means that the rheological properties are changing dramatically, but the structure of the polymer network actually is pretty conserved. So if you think about the entangled solution at the high temperature or fully gelled solution at the low temperature, yes, they are totally different from rheological perspective, but structurally they are almost identical. So these are lookalike molecules with lookalike gel structures, but markedly different rheological properties. What is the underpinning mechanism? So we started looking at, of course, because the linkage composition is the same, started looking at the motif and the degree of the substitution. And we could really got the inspiration from the work by Paul Dupré, that he looked at the two-turn twist and three-turn twist motifs in the xylans that he extracted from the arabidopsis, and we performed these optical rotary dispersion, which is the circular diachroism for non-absorbing molecule, and we identified that there are differences in the secondary structures that are actually dependent on the temperature. So the secondary structure of the chains is changing as a function of temperature. And so we took again this work by Paul Dupré's group, who identified this conformation by the double quantum NMR. We didn't have access to the double quantum NMR simply because we couldn't produce our polysaccharide in the C13 enriched atmosphere, which is something that we still try to do. But we could analyze the properties of this fraction using the one-dimensional NMR, and knowing where to look for the peaks, thanks to the work by Paul Dupré, we could identify that actually the water fraction has a threefold screw, so it's more chaotic, and the alkaline soluble fraction is a strictly twofold screw molecule. And you can visualize those differences in the secondary structure using atomic force microscopy. We also collaborated with Elena Wolf in the Queensland University of Technology, who looked at us at this fraction using the helium ion beam imaging, and you can see differences in the secondary structure and molecular arrangement. And finally, we collaborated with the University of St. Andrews and Basque Center for Applied Mathematics and our PhD student Peter Stars to look at essentially modeling these polysaccharides and looking at the information that we can get about why these changes in the secondary structures are occurring and how the different substitutions are impacting the secondary structure. So what is the role of the side chain on assembly and the secondary structure? So we did a very simple enzymatic treatment of these polysaccharides. We cleaved terminal arabinoses using arabinofuranozidase because most of the xylanases actually are not hydrolyzing these polymers because of the high level of the substitution. So in this case, we're using the arabinofuranozidase to treat these polysaccharides. And essentially what we see is a huge differences in the water fraction that undergoes a transition from a gel to liquid once you pull out these terminal arabinoses. For the alkaline fraction, the enzyme is still active. We can quantify the release of almost 100% cleavage of terminal arabinoses, but it has no or very minimal effect on the rheological properties. And the same we kind of proven in terms of the changes in the secondary structure as a function of the cleavage. And we could see that the water-soluble fraction undergoes transition in the secondary structure. It loses the twist screw, it becomes more random, and it becomes fully dissolved, loses its gel properties. But for the alkaline fraction, we couldn't find that. So final thing that we would like to mention is the interaction of these things with water. So the thermodynamics of the interaction of these things with water is very different. The amount of the bound water is very different between these two fractions. And the mobility of water in this bound state is very different, which we associate with the different motif of the side chain distribution. And that leads us to a hypothesis of what is the gelling mechanism and why it is so different to many other polysaccharides. So we all know about this hydrophobic effect, where the surfactant molecules that made of hydrophilic and the hydrophobic tail, they aggregate if the concentration exceeds a certain value, which we call the critical micelle concentration. And we know that the thermodynamics of this process is such that it's not driven because the chain of hydrophobic chain want to aggregate. Hydrophobic chain don't want to aggregate. It is water molecule that are not happy, and here is the unhappy faces, they're not happy around these hydrophobic chains. And that results in the huge entropic gain when these chain aggregate, and this unhappy water molecule join the bulk, the pairs in the bulk, and gain the happiness, and that results in the much higher levels of the entropy when these molecules in the bulk, in comparison with the very low entropy level when these molecules are adjacent to these hydrophobic things. So we have this hypothesis about how these polysaccharide interacting, taking this information from the DSC data and the relaxometry data using the proton relaxation. We have the hypothesis that obviously the water is associated with this polysaccharide due to the enthalpic gains, but entropically it is very unhappy. And when you have association in this case, you still have retention of water. So unlike most of the junction zone that go with the expulsion of water, like for example, cellulose or starch, when the starch or cellulose molecules interacting, usually the water goes all together. Here, the water is retained in the junction zone. So think about the pectin, where it is the ions of calcium that act as a crosslink. Here is the water acts as the crosslink. And of course, the number of water molecules in this case are entropically in the absolutely dead state. They are really sort of unhappy entropically. But because you release some of these water molecules from the original two chains into the bulk, the happiness of these molecules that released outweighs the negative impact of the confinement of these lubricating water molecules that form these junction between polysaccharides. So this is the hypothesis that we are currently working on to prove exactly the state of these molecules. And overall, because these guys are very unhappy, but these ones are happy, the overall is a positive. It's much better than having a negative overall, slightly negative, but altogether it's a negative, and altogether here is much more positive. So final minute before we go to questions, can we do the reverse engineering? So can we embed now these properties that are unique to xylem to other polysaccharides? Of course, there's a work in progress. So we have a collaboration with Professor Wei Zheng in the Zhejiang Agriculture and Forestry University, where he develops these GT61 xylosyl transferases from Plantago ovata that specifically decorate the chains with the 1,3 xyloses. And we can take a wheat arabinoxylan and create a de novo, produce the decorated xylan. In our lab, we are doing the characterization of the physical properties. In this case, we use the quartz crystal microbalance because we had a very small amount, so we couldn't do the full-scale rheology, but we could show the differences in the adsorption and relaxation properties using the quartz crystal microbalance. So on that, I just would like to thank all the collaborators, all the funders, and thank you very much for your attention.